Okay, so I think we'll make a start. My name's John Draper. I'm the Program Director for Research and Partnerships at the Stem Cell Network. Um, so welcome everyone to the first presentation in the Stem Cells from the Sofa speaker series. This is a virtual seminar series that is designed to reconnect researchers and bring expert content directly to your sofa. The series will feature a different speaker every 2 weeks, and the talks will run for about 20 minutes with 10 minutes of questions at the end. So I would now like to introduce our first speaker in the Stem Cells from the Sofa speaker series. It's Dr. Michael Rudnicki. He's a senior scientist and director of the Regenerative Medicine Program at the Ottawa Hospital Research Institute and scientific director of the Stem Cell Network. Michael is an internationally recognized thought leader in molecular genetics and regenerative medicine, whose research has transformed our understanding of muscle development and regeneration and fueled the development of novel molecular and stem cell-based approaches to the treatment of muscular dystrophy and other neuromuscular disorders. Today, Michael will be talking about the molecular regeneration of stem cell function. Please take it away, Michael.
Thank you very much, John. I'll just have to grab the screen. Okay, I hope everybody can see that. So first of all, it's a real pleasure to be the opening speaker for this series, and I think the series is going to be a lot of fun. And thank you to John Draper for putting this together and for the invitation to lead off the series. I was asked to speak to our efforts around commercialization and explain some of the path that we took to translate our findings via the commercialization route. And so my first slide really is a disclosure that I'm a founding scientist in Satellos Bioscience. This company now is about 2 years old. We've really been up and running about a year and a half. And the different elements that really are important for one to take this kind of path are, well, for me, it was having a colleague and friend, Frank Gleason, where we have a lot of trust and respect for each other. We actually first met when he was a board member of the Stem Cell Network, was the chair of the board for a while. And he's an entrepreneur who's been involved with multiple biotech startups. Before that, he was in the venture side, very experienced, very knowledgeable of the space. And that really, having a connection on the business side, of course, really helps facilitate things. Having the support of your tech transfer office and having a good relationship with your tech transfer office is also very important. And for me, our person is Anouk Fortin. She's really been very amazing and very supportive in advising on patent strategy and helping support those patents, especially before we had money in the door. I think also our institute has been very supportive overall, which is also helpful. And also having an investor, especially at the seed stage, is really key. So my thank you to Bloom Burton, the investment bank that has supported this venture. Their support and other things has really been essential, and their network as we pursue Series A funding has been essential. So there's multiple elements to make things happen, and of course the science has to be there and has to be ripe for translation. Let's move on to the next slide. So I'm talking about muscle stem cells. These are also known as satellite cells. Skeletal muscle are your voluntary muscles. These are the muscles that allow you to move around. We're not talking about heart or smooth muscle. Your muscles are groups of muscle fibers which extend from the whole length of the muscle. They are a syncytium that are formed through fusion of progenitors or myoblasts, and they contain thousands of nuclei. Around these fibers is a basal lamina, and between the basal lamina and the fiber is a population of cells called satellite cells, and these subset of, we believe, are the stem cell compartment of these cells. These cells give rise to progenitors through asymmetric divisions, and those progenitors can divide multiple times before differentiating and maintaining to maintain and grow and repair the tissue, muscle tissue. And muscle is extremely regenerative, as those of you who've climbed up or down a mountain or exercise heavily know. And there's a number of markers that identify satellite cells, and PAC7, a transcription factor that we identified, is expressed in the nucleus of satellite cells, and there's an example of satellite cell beneath the basal lamina in green. Now, satellite cells and muscles, satellite stem cells in particular, can divide in different ways. They can divide symmetrically. They give rise to two stem cells, expanding the stem cell number. The division is oriented in a manner parallel to the basal lamina. The basal lamina is on top of these cells, and that is termed a planar cell division. When an asymmetric division occurs, it occurs at 90 degrees to the basal lamina. The basal lamina is against the stem cell, and the cell that's committed to become a progenitor is pushed into the muscle fiber, which then can allow either for direct fusion and differentiation, or it can also proliferate under conditions of more acute injury. And that leaves behind one stem cell, so we're maintaining the numbers of stem cells. We can also have that stem cell loss through direct differentiation, which may or may not differentiate, leading to stem cell exhaustion. And we've come to appreciate over the years that the balance between symmetric and asymmetric division significantly impacts the efficiency of the regeneration process, and also that it is a regulated process with multiple signaling pathways impinging upon that decision whether to do a planar or apical basal asymmetric cell division. And hence, it's also druggable. The disease that we're have a keen interest in is Duchenne muscular dystrophy. This is the most common form of muscular dystrophy because it's a very large gene located on the X chromosome, so you only need one hit, and a large proportion of them are new hits, but they're also inherited. It's an awful disease with extensive muscle wasting throughout the body, an inability to walk by around 8 years of age, and death typically in the second decade of life, although some of them nowadays with the best medical care make it out to their early 30s. The disease gene is called dystrophin. It's part of a large protein complex that sits up the membrane of differentiated myofibers, and loss of dystrophin destabilizes the complex and it's lost. And the function of this complex, you really can think of it as a rivet joining the outside to the inside of the muscle fiber, providing structural integrity. When this complex is lost due to loss of dystrophin, one has lots of membrane breaks that are long-lasting and myofiber damage and death, leading to bouts of degeneration and regeneration, extensive fibrosis, as you can see between these muscle fibers, and fatty infiltration. There are, I think, in recent years, great hope raised around gene therapy approaches using AAV vectors and even gene correction using CRISPR-Cas. AAV, however, does not efficiently infect satellite cells and really targets the myofibers, so the satellite cells are not being helped. So we noted, and I think made a very important seminal contribution to this field a number of years ago in 2015 in a Nature Medicine paper, where we noted that asymmetric division involves dystrophin and the DGC complex, where dystrophin in muscle stem cells has a novel signaling role. It accumulates at the basal cortex, the north pole, recruits a kinase called PAR1B that prevents the assembly of the PAR complex, which instead assembles at the opposite cortex, polarizing the cell, recruiting centrosomes, allowing a mitotic spindle to form at 90 degrees to the basal lamina, and for an asymmetric cell stem cell division to proceed, giving rise to a cell that will become a progenitor. In DMD, there is no dystrophin, there's no, PAR1B is not recruited. The PAR complex sets up around the cell. We have evidence for multipolarity leading to mitotic catastrophe and the loss of these cells through programmed senescence. Some cells do squeak through by chance, but we see a large reduction in the proportion of cells being able to efficiently undergo asymmetric division. When asymmetric divisions do occur, the progenitors that are made are pretty normal. They have some issues, but they're pretty normal. And they're able to grow and maintain the tissues at a certain level. However, the flux from the stem cell compartment to the progenitor compartment is vastly reduced. There's a stem cell hyperplasia in DMD. There's a 3 to 5-fold increase in PAX7 positive cells at all stages of the disease in both humans and mice, and a deficit in the numbers of progenitors is marked by a reduced proportion of myogenin positive cells. So we have a stem cell hyperplasia and an inability, a reduced ability to flux and generate progenitors. And we hypothesize that this reduced ability contributes to disease progression in a significant way in DMD. And the evidence in support of that, I think, is quite dramatic. If one deletes either DAG1, which is a transmembrane portion of the complex, and we've also done this with dystrophin, but it's not published. If you delete only in myofibers using a differentiation-specific Cree driver, you have a very mild phenotype and normal regeneration. If you delete it in satellite cells, you have the progressive normal muscular dystrophy. So the progressive aggressive DMD requires two things: ongoing muscle damage in muscle fibers, together with a reduced ability to repair it. Also evidence that regeneration is, ameliorating regeneration is really key to ameliorating the disease phenotype. Comes from a number of different sources, including identification of escaper dogs in a DMD dog, the full DMD deletion, but they had upregulation of JAGED1, which is, of course, the notch ligand and affects the regeneration and polarity process, restoring regeneration of normals. And this was recapitulated using zebrafish. There's also escaper DMD patients that don't have modifications in known modifiers. This 15-year-old patient has a very mild phenotype, no dystrophin, pretty normal looking muscle, whereas his half-brother, who's 9, is in a wheelchair and has the full-on DMD phenotype. So you don't need dystrophin protein to get better. You need to have enhanced regeneration or rescued regeneration. Okay, so in parallel studies, we identified a novel signaling pathway that enforces polarity in these cells. This is a novel form of the EGFR pathway, where EGFR, when activated, recruits Aurora kinase A, which then phosphorylates PAR-D3 and knocks it out of the PAR complex. EGFR is located at the basal cortex, just like dystrophin, and functions perfectly normally in the absence of dystrophin. And the way I think of it is that the dystrophin pathway sets up the default levels of asymmetric division, because the EGFR pathway allows that to be regulated either plus or minus through activation or inhibition. And we published a paper in Cell Stem Cell last year where we extensively resected this pathway and showed that agonizing this pathway drives asymmetric division, and doing that in MDX mice rescued the loss of asymmetric division and restored the phenotype to near normal. This was done through electroporating EGF expression vectors into an individual muscle in the leg. So we're thinking about how we could drug this pathway. We don't want to turn on EGFR willy-nilly. We want to do it in a context-specific way where it's normally being activated. And so after some thought and much experimentation, we identified a protein tyrosine phosphatase. It's codenamed here PTPX, because this is our secret sauce at this point. This is highly expressed in muscle stem cells. And its normal role is to turn off the EGFR once phosphorylated on tyrosine once it's been activated. And normally if you signal through EGFR, it's a spike of phosphorylation on EGFR. It goes on and off. And that off uh, um, being turned off is facilitated by PTPs. And there's usually multiple ones that do this, but not in muscle stem cells, interestingly enough. And we thought that if we could inhibit this PTP, we could prolong activation of EGFR and get the same end result, the asymmetric division, by essentially not agonizing the pathway directly, but by keeping the signal on longer. And that's indeed exactly what happens. And through a lot of studies that I'm not going to tell you, we've developed a allosteric inhibitor that doesn't bind the enzymatic site of PTPX, but rather binds a fold in the back of it that then inhibits the conformational changes required for it to be active. And this inhibitor absolutely drives asymmetric division, both in our in vitro assays and in vivo, and really has a remarkable effect on MDX mice, which I'll show you in a moment. So Satellos really has spent a lot of time and effort on generating drugs that inhibit PTPX. We have undertaken multiple approaches, including fragment screening, virtual screening, and have identified 5 lead series. And in September of last year, we picked one of these to do an in vivo proof of concept study. And we call that SAT-732. And the design of that study was really, we were looking for a signal. We picked a low dose, 5 mg per kg, that were delivered 3 times per week by intraperitoneal injection. Our reasoning is that this dose was picked based on our PK/PD studies that suggested this gave us enough drug to give us a reasonable inhibition of our target. And we also did it intermittently because we hypothesized that pulsatile treatment would facilitate the cycles of regeneration that normally occur, and that may well be better than continual inhibition, analogous to exercise regimes, and so on. So we took very young MDX mice, the mouse model for DMD, and we treated them for 4 weeks, from 6 weeks of age to 10 weeks of age, getting 3 injections per week. They were given a running wheel that was monitored via Bluetooth, uploading its data every minute for physical therapy, and also to see how they're doing in live time. And at the end of the experiment, we conducted force measurements, histology, and morphometric analysis. And we were really just looking for a signal. And I must admit, we were quite amazed by what we saw with this, essentially what is a tool compound. We're not yet at our clinical lead. This is something on the path to a clinical lead. So first of all, we saw, and this is showing you the diaphragm, we see essentially the same thing in the diaphragm. The numbers of PAX7 positive cells is reduced towards normal levels, and the numbers of myogenin positive cells is increased towards normal. Now, PAX7 marks all satellite cells and progenitors, and myogenin marks a progenitor that's committed to differentiate. We don't have a good marker for progenitors only, but what we know is that in DMD patients and in MDX mice, the proportion of myogenin positive cells in tissue is abnormally low. And so what this shows is that we're seeing a decreased stem cell and increased progenitors, and provides a really elegant proof of mechanism of action of the drug. We're on mechanism. One of the things we noticed right away is that the mice ran more. Mice love to run, and drug-treated mice ran 23% more than MDX mice. These are pretty young mice, and the dystrophic phenotype hasn't really fully manifested yet, so they're still running quite a bit. I don't know how many of you can run 200 or 160 kilometers in that period of time. Certainly I can't, but this increase compares quite favorably to the difference between wild type and MDX mice, which is about a 25% difference in freewheel running for this age of cohort. This really suggests that the drug is doing something in a positive way, and they're certainly not fatiguing. They're running faster, and they're running longer. So the drug is really having a positive effect. We also saw a really impressive effect on titanic force generation in the tibialis anterior muscle, 14% increase in force. And again, this is comparable to approaching that of wild type. And we know from historical experiments, this is comparable also to microdystrophin infected muscles. So again, simply by enhancing regeneration, we're having quite an impressive effect on these performance of these muscles throughout the body. This is the tibialis anterior muscle out of the lower leg, and histological appearance of the control versus drug-treated animals. Just looking at the center in the laminin, you can see the thickness of the red here, and the control is quite a bit thicker than that of the drug-treated. In fact, the drug-treated looks essentially like normal muscle. You have these necrotic areas with many small fibers that are wheat germ agglutinin staining, which is evidence of fibrosis that you don't see in the drug-treated animals. Really quite a remarkable change in the histological appearance of the muscle. And if we look at this, we can see that there's a 33% decrease in fibrosis by WGA staining, and a return of the caliber size to a more uniform size, and without these very small fibers that are evidence of an attempt at regeneration in these necrotic areas or damaged areas, focal areas of damage. And similarly in the diaphragm, you can see this difference in the thickness of the diaphragm between drug-treated and the control. You have these fibrotic areas that are void of muscle fibers that just aren't present, more uniform fiber ferret size. So again, we have loss of these ultra small fibers, and a more normal distribution of muscle fibers, and a big decrease in fibrosis. So remarkable effect on the diaphragm. And we see no evidence for toxicity of the drug by weight of the animals. And also on necropsy, we saw nothing abnormal. So this is encouraging that we're not doing something awful to the animals with this drug. They're eating well, they love to run, they seem pretty happy. So this just summarizes what we found. Really quite a remarkable effect of a drug that has yet to be gone through this final optimization. It's being delivered by IP, which mice don't like. I can tell you, giving that 3 times a week doesn't, they're not happy about that. We, of course, we're working to have it orally available, and we think this is a really exciting development and points to a novel therapeutic that will complement multiple other treatments, including gene therapy. And by restoring the regenerative potential to, we hope, normal levels, this really is a novel strategy that addresses a mechanism of disease that hasn't been addressed in any other previous therapy. And I think I'm even 5 minutes early, but here's the crew that did the job, and I'm happy to entertain questions.
So, Michael, you have one question. Can you briefly cover how autophagy functions in muscle stem cell function and differentiation, and possibly in disease states?
Yeah, well, there's multiple avenues that we have not evaluated. This is our first POC experiment, and we really would like to do multiple experiments to fully analyze these animals, including longer-term treatment of older animals, combination with gene therapy, as well assessing metabolic aspects, mitochondria, what's going on with inflammation, what's going on with autophagy, um, uh, and whether or not those particular deficits that we know are there in MDX mice are ameliorated or not. And we just haven't done that yet. But that's a great question, Colin. Colin is also asking whether we would see depletion of the muscle stem cell pool. We have not seen that. So the increase in asymmetric division isn't occurring, I think, at the expense of normal levels of symmetric division, and an asymmetric division leaves behind a stem cell, and so we're not decreasing symmetric expansion to very low levels, and so we think that mechanism, together with treating intermittently, would obviate or not lead down that path. So we don't think that's going to be an issue. Of course, we have to do a long-term treatment. However, if we have, let's say, an accelerated aging phenotype, and people run into problems after leading 10 years of normal life, that might not be a bad thing. And perhaps one would then slow down the treatment if that was an issue. But it seems that the way these controls work is that they tip the balance, but they don't overshoot. So we hypothesize that there's feedback mechanisms. Certainly in wild type mice, when we treat them with electroporation of EGF, we have minimal effect. And we don't make muscles bigger or alter the stem cell compartment size. Let's see, from Anonymous, was specific function improved? Also, any change in CNS? I'm not too sure what you mean by specific function. Do you mean the quality of the muscle, normalized muscle? The normalized muscle was somewhat improved, but we are looking here at total force because, at least in my opinion, the total force of the muscle is what you are dealing with as a patient. You have your whole muscle. So if you normalize to volume, and you have a small, stronger muscle, your total force may not be stronger, if that makes sense. I don't know what you mean by CNF. From Mayank. Yes, asymmetric division does play a role in aging. And what happens in older animals is that rather than having reduced asymmetric division, you have reduced symmetric division. And we published a paper that showed that there is a growing signaling through the JAK2 STAT3 pathways as one ages, and that by inhibiting that, you could restore symmetric division to normal levels in aged mice in the context of acute injury, and also in engraftment assays, restoring the function of stem cells to normal levels in an aging context. So we have different strategies for stimulating symmetric division, and we're interested in evaluating those in a aging context. So one can manipulate the stem cell to make either undergo more symmetric or more asymmetric division. Let's see, Penny. Yeah, why did we select this one? In September, it was our best one. So the question was we noted several lead molecules, and why did we select the particular one that we moved forward with for the in vivo studies? So we wanted to get some in vivo POC data in the bag That was also important from a investor relations point of view, to wave the flag that what we're doing makes sense and is exciting. And so in September, we just picked the best one, the one that had the highest affinity, the better specificity, and so on. Now we have others that are better, and that's continuing through multiple cycles of medicinal chemistry. So it's a bit of a horse race, if I could put it that way. From Anonymous, what are your thoughts on the truncated version of EGFR to inhibit the downstream signaling that is used in the context of CAR T technology? Is it transferable to the muscle stem cell context? So are you talking about truncated versions that are oncogenic, that are constitutively active, or are you talking about a dominant negative form? I'm not too sure what you're referring to. So what you'll have to explain a little more about the EGFR. Certainly, to introduce a mutation in EGFR is analogous to gene therapy, and so one would have to use cell therapy. Doesn't work for muscle. That's been relatively well established, although there's still some people trying to work on that, and with more or less success. Gene therapy approaches really aren't very effective at targeting the satellite cells, so I don't think that would work. Do you think drug delivery was affected with the exercise? We just don't know that. We haven't done PK studies through exercise. And would we expect similar results without running wheel? We haven't done that experiment. But that's a good question. I think the experience of many groups who've targeted muscle is that where things work best with getting off the sofa. If I can say that, a little exercise helps. So I would, movement is a good thing, and in DMD patients, they do a PT also to prevent seizing of the muscle and to keep their mobility going. So I think that some kind of exercise will be beneficial. I don't know how it affects the response to drug, and that's a good question. These satellite cells are already activated due to muscle damage. My personal view is that this is helpful. Francis, yes, is PTPX expressed in other tissues? That's a great question, and we're also very interested in that. That'll be part of our talks workup. It is widely expressed at low levels. It is highly expressed in muscle stem cells. It's typically expressed as part of with multiple PTPs that have the same targets. So there's a lot of functional redundancy, those other redundant members of the family are not expressed at very high levels of muscle stem cells for some reason, allowing us a certain degree of specificity. So it is possible that we'll see activation in other places, and we're going to need to assess whether that's going to be an issue or not. We do know that relatively low levels of inhibition, something less than 50%, gives us the full biological effect, and that together with low dosing, intermittent dosing, we think will address these possible issues. We did not see any adverse effects, at least after 4 weeks of treatment, but of course this is something that will have to be fully addressed in time. How different are the regeneration pattern between wild type and MDX mice? So we did not do a cardiotoxin injury, if that's what you're referring to, with SAT732. We thought that it was more physiological just to give ongoing treatment to a normal mice. With other tool compounds, we have done CTX injuries, earlier versions that were lack specificity, and that did appear to enhance regeneration in MDX mice. Do I have a positive control? If so, what happened to that group? Eric, if you're referring to wild type mice, we did not do wild type cohort in this experiment, and we will be doing that from now on in. It was a shooting from the hip kind of experiment that we're doing quickly. So yes, indeed, we need a wild type cohort, if that's what you're referring to as a positive control. Um, Gene, myogen indicate commitment. Yes, in this experiment, we did not look at proliferation. We did that extensively with EGF electroporation. Now, EGF is a well-known mitogen. In this case, in skeletal muscle, it is not. We saw no effect by Ki67, EDU, etc. The only thing we seemed to affect was satellite cells and satellite cell asymmetric division. And John, am I out of time? We have time for more questions. What time is it?
Why don't you finish, do one more.
Okay, reduced fibrosis. Yeah, okay, so the question is whether we're targeting FAPs. I think that the explanation is, and it will require some study to fully address this, but I think by restoring the generation of progenitors to normal, these are dividing and expanding, presumably. And from Fabio Rossi's work, I understand that progenitor expansion induces a die-off of FAPs. So I think that's an attractive mechanism to consider, and that it would be just the balance between FAPs and progenitors and their normal interactions are restored, leading to restoration of FAPs to normal levels in a post-regeneration context.